Carrots are biennial members of the umbel family, domesticated between 900 and 1700 AD. Historical evidence suggests their first point of domestication was in modern-day Afghanistan, with other potential locations in northern Iran and Pakistan. Carrots are known around the world as a nutritious root vegetable that contains high concentrations of vitamin A, antioxidants like beta-carotene, and other beneficial phytonutrients.
In order to gain a perspective into the current state of modern carrot breeding, Dr. Irwin Goldman, a respected carrot breeder from the University of Wisconsin-Madison, was contacted and interviewed for his expertise on the subject.
According to Dr. Goldman, one difficult aspect of carrot breeding is that it takes 1 full year to produce a new generation of usable seed. In a typical breeding cycle, the crop is harvested from the field after the first growing season, then the root goes through a vernalization process in a controlled environment, followed by the use of hybridization to increase seed production and make new genetic strains. Additional non-breeding specific issues associated with carrot production include pests and diseases such as Alternaria douchi, the carrot leaf blight, Cercospora carotae, the Cercospora leaf spot, Silo rosae, the carrot fly, and root knot nematodes in the Meloidigina genus. These pests and diseases are yield limiting and continue to challenge breeders due to the lack of resistant carrot varieties. In the current market, breeders focus on improving traits such as color, sweetness, flavor, root uniformity, and disease resistance. And thus far, major improvements have been made on yield, shortening growing period, and changing the root color. For example, did you know that early carrots were yellow, white, red, and purple? The iconic orange color was actually selected for in the 17th century as a tribute to William of Orange, a monarch and major proponent for Dutch independence. The color orange was a part of the Dutch independence flag, and this color of carrot has remained a favorite for over 300 years. Root shape has also been the target of extensive selection, leading to carrot varieties of multiple shapes and sizes, known as market classes. Carrot breeding has a diverse set of market classes that must be taken into consideration before starting a program. One major factor in determining a carrot's market class is end-user interaction. Carrots for the fresh market must be aesthetically appealing in terms of color and shape, while also full of flavor. Carrots for the processed market must maintain acceptable flavor, but are also bred for bulk and tonnage rather than aesthetic traits. The 4 major market classes of carrots are Chantenay, Danver, Imperator, and Nantes. Chantenay carrots are short, have a large girth, and a rounded tip. They store well and are mainly used for processing. Danver carrots are conical in shape with a sharp tapered end. They are shorter than Imperator carrots but manage better in heavier soils and are typically used for both processing and fresh market production. In the United States, the most common market class is Imperator, which features a deep orange color, a long tapered root, high sugar content, and is grown predominantly for the fresh market. Finally, there is the Nantes carrot, which features a round base and is slightly larger than the Chantenay. Its primary use is in the fresh market as well. Dr. Goldman has stated that each class has unique characteristics that breeders must adhere to, but that market classes are, quote, somewhat malleable, and that it is possible to breed between different market classes over the course of a few carrot generations. Many major carrot breeding programs depend on utilizing inbred lines and cytoplasmic male sterile plants to create vigorous hybrid offspring. However, carrots are highly sensitive to inbreeding depression, so carrot breeders must walk a fine line when dealing with the traits of inbred carrots. Despite the risks, inbred carrot lines are commonly used for their ability to generate large amounts of seed in the field, create quality F1 hybrids, and produce sterility in the female line. This project would like to thank Dr. Irwin Goldman of the University of Wisconsin for giving us our time in the interview, and would also like to thank Kellen Andrea for his voiceover work in the guest paragraph.